All right, folks. So something where people get confused, and maybe you can test yourself to see how confused you are. So here is a cell with a chromosome there. So what I want you to ask is how many chromosomes are shown right now? Sorry, let me make this look better. Are shown right now versus right now. So again, we started here and now here. How many chromosomes? Well, to answer this question, it might help to think about this. So first off, let's start with this cell. And let's say this cell is at the beginning of interphase. So the first thing the cell's got to do, it's got to go through G phase. After it goes through G phase, G1, I should have said. So it goes through G1, then it goes to S phase, and something happens in S phase. A lot of you can probably answer what happens in S phase is that the DNA gets duplicated, right? So here's my chromosome now. So now I'm in S phase. And then I end up going from S phase to G2. Okay, my cell grows more. And then once I'm in G2, my cell is going to go under mitosis. So it'll go to M phase or mitosis. Now in mitosis, there's a few different steps that happen, right? The chromosomes condense, so now they look like chromosomes. Then they go to the middle of the cell in metaphase. After metaphase, the spindle fibers attach, they separate the chromosome, or they separate the sister chromatids, I should say. And then from there, the cell splits in half, and then it's ready to do the whole thing over again, right? It can copy the chromosome in this cell. It can copy the one in the other cell. So if we go back to that original question, which you could either go back or I'll put it right here again, how many chromosomes are shown here versus here? Well, really, this is one chromosome shown right here. And when it goes through S phase and duplicates, this is still considered one chromosome. Because the way you count chromosomes is actually by the number of centromeres. And here there is one centromere. Here there's still one centromere. So this is one chromosome. This is still one chromosome, but it is two sister chromatids. So there's two sister chromatids shown there. They're identical, but it's still considered one chromosome. Okay, so that was one part of confusion. Okay, the next part of confusion is, okay, now we're done talking about mitosis, and we're going to talk about meiosis. And during meiosis, I have all of these chromosomes, and these chromosomes are paired up. So there's these two chromosomes, there's these two chromosomes, for a total of 4. Now, before meiosis happens, we'll assume this is a body cell or any cell, really. I guess technically meiosis can only happen in a sex cell. So this would only be happening in sex organs. And these chromosomes, they align differently. So in metaphase 1, my homologous pairs line up. But before that, my homologous pairs, which have the same genes on them, right? So these two up here might represent genes for hair color. So maybe that shows hair color, and these have genes for hair color. And maybe these have eye color, and these two chromosomes are representing eye color. So in prophase, they share pieces with each other. So I'm going to demonstrate that by ripping a piece off and showing that. So here is that. Here's this. Get it? There we go. Okay, so crossing over, we have seen has happened in this cell. So they've crossed over. Now in metaphase 1, the chromosomes line up in the middle like this. And they could either line up in this orientation, or they could line up in this orientation. Right? Subtle flip. So they just can flip-flop spaces. That process there of lining up in different spots is known as independent assortments. So this is meaning, if I'm talking about these as probabilities, they're each of these probabilities is equally likely to happen. So there's a 50-50 shot that they'll line up that way or that way. Why is that important? Well, in meiosis 1, these chromosomes are going to get pulled apart. So I'll show that here. So these chromosomes will get pulled this way. These ones will get pulled this way and vice versa. The cell is going to split down the middle. And my cell could either look like this, or we could have imagined a scenario, right, where they lined up like this as homologous pairs. So these are homologous pairs because they have the same genes on them. But they could have also lined up like this, right? And so when they split in meiosis 1, I could end up with a couple different variations of what this cell could look like, these two cells. That's a key part of where genetic variation comes from, besides crossing over. Okay. Now, when I get to meiosis 2, so now I have two fully different cells. Okay, so here's the cells that underwent meiosis 1, and then they're going to go into meiosis 2. So now they're entering meiosis 2, where these chromosomes will then line up along the metaphase plate. But this time, again, the homologous pairs have already been split apart. So now we're going to actually start splitting apart the sister chromatids. If you paused the video and went back to before we split these cells, you'll notice that the number of chromosomes in the original cell was 4. There were 4 chromosomes. There was 1, 2, 3, 4 that made up the original cell. When they split, now I have 2. So I have half as many chromosomes per cell as the original cell. This is why these are now haploid. And now we're going to split them again, but this time these sister chromatids split apart. So they're going to split like this. There they go. And they split. It'll go right down the middle. I'm going to redraw these. But before I redraw these, we can imagine, again, these lined up in the middle at metaphase 2. When these are lined up like this, they have two ways that these chromosomes can line up, just like before. So this time, these chromosomes could be lined up like this, or it's equally likely that it lines up like that. So this way or the other way. Same with this one, this way or the other way. So like that or like that. Because of this, this can lead to, again, this is another example of independent, which I won't write the full thing, assortments. So these, again, equally likely probabilities of either of these orientations lining up. And when we show these cells splitting, so when they do split into two separate cells for each of them, either of those cells is equally likely this way or this way. And for both, right? Either one could do that. Because of that, after meiosis 2, we end up with 4 total, right? We should have one cell. There's one cell here, one cell here, one cell there, one cell there. So 4 cells, and they're going to stay haploid. Why? Well, we said these cells were haploid, like that, because there's 2 chromosomes per cell. Well, remember we said at the very beginning of the video that even just one of these counts as a chromosome. So when these sister chromatids separate, there are still only 2 chromosomes per cell. So each of these cells still only has 2 chromosomes. Therefore, we haven't halved the half, or another way to think about that, we haven't taken the haploid number and divided it by 2 again. It stays haploid. So just a quick summary of these events. So as you can see on this image, this was showing the same type of setup where we have 4 chromosomes. So here are 4 chromosomes in this cell, right? You can see them here. There's 4 chromosomes. There's also 4 in here. And here they've split. So each of these cells now has 2 chromosomes, 2 chromosomes. And again, these are showing 2 options. So we're going to ignore this bottom scenario for now. This is just if independent assortments happen. We're just going to focus on this. So again, we have 2 cells with 2 chromosomes each. These cells are going to split. Again, they're going to split in half down the middle of each of them, right? Each of these sister chromatids is going to get separated. So we still have 4 cells with 2 chromosomes each. So here, these are both haploid, and this would be diploid. Or another way to think about that is this would be 2N, and each of these would be equal to N. Okay, hopefully that helped with independent assortments, diploid, haploid, and we'll catch you later.